Hey, it's Mr. Bonney here with another biology lesson. We're looking at genetics and some of the basics of genetics. So let's first just start off with the very root of this, which is what a gene is. And a gene is just a section of DNA that has the information to code for one protein. And we also refer to genes as traits. So an equivalent that I oftentimes write down and think about is that one gene is equal to one trait, which is then equal to one protein. When we look at it from a protein synthesis point of view, the DNA code necessary to make one protein is a gene. And then we also refer to that as a trait. Now within traits, we also have something that are called alleles. An allele is just a version of a trait. So let's say, for example, that we have a trait, and that trait is height. And let's say it's the height of a pea plant, how tall it can grow. That is the trait. The alleles then for that trait would then be, say, tall or short. These are the two options that can be represented by that trait. Those are my alleles. Now those alleles get represented in genetics by a shorthand, and we typically find the dominant trait and we use that as our letter. So for then tall, I would represent that as a capital T, and then short, I would represent that as a lowercase t. So in a dominant trait, it is the one that will win out when it is present, and also the lowercase or the short trait is present, which then gets us to genotypes. A genotype is all of the letters that someone has for the trait. So for height, there's three possible genotypes, which would be capital T, capital T, capital T, lowercase t, or lowercase t, lowercase t. What's important to remember with the genotype is that each individual has two copies of each trait, one of which that they got from their mom and the other of which that they got from their dad. The genotype then controls what we call the phenotype, or how the individual actually looks. So both the capital T, and capital T, lowercase t make a pea plant that is tall, whereas the lowercase t makes a pea plant that is then short. That's the then phenotype of these plants. The last thing to talk about is expanding upon the genotypes. Genotypes can be either homozygous or they can be heterozygous. So a homozygous genotype is one that has two of the same letters. So it can be homozygous dominant, and that would be capital T, or it can be homozygous recessive, which would be lowercase t, lowercase t. A heterozygous then genotype would be one capital and one lower. They're two different traits. Now heterozygous in this case is always dominant because the dominant trait will then win out over the recessive trait. Okay, let's take a look at how to do a Punnett square. And before we dive into how to do the Punnett square, let's remind ourselves of what's the point of a Punnett square. The point of a Punnett square is to take a look at two individuals, looking at their traits, and then see what their kids would look like based upon the traits of the parents. Now, in this type of Punnett square, we call it a monohybrid, or a single trait that we're going to look at. So in our fictitious Costa Rican mushroom-hugging tree frog example, we have a few traits over here. We have the trait for the color of them. They can either be red or orange, where red is dominant, orange is recessive. They can either have 5 toes or 6 toes. 5 is dominant over 6. And their eyes can either be bulging or flat, where bulging is dominant over flat. So in this first problem here, let's take a look at the setup for it. They tell me that I have a 6-toed female. Now, that's a number of toes. 6 is the recessive trait. So the only way that I can have a parent with 6 toes is if that parent has 2 lower cases for that trait. Remember that each individual has 2 copies of each gene, one that they got from their mom and one that they got from their dad. Then the male, they tell me, is heterozygous. Now, heterozygous, as you remember, means different. So that means this parent would have one capital letter, so one dominant trait, and one lowercase, so capital lower. So these are the traits of the parents. Now, what I need to look at is what happens when they have kids. So remember that each parent will donate just one copy of their traits to their kids. So essentially what I want to do is I want to take these traits and I want to split them up. So I've color-coded it for you, but I'm going to take that first trait there and put it there on the left side of the Punnett square, and the other one here. So the first parent, the one that I've highlighted in red, their traits will get split up on the left-hand side. The second parent, I'm going to split them up along the top. Then the purple boxes in the middle are going to be the intersection of these two parents. So for example, if the blue parent donates this capital F and the red parent donates this lowercase f, this would be that product of their kid here, which would be capital F, lowercase f. And again, I look up and I look left every time I fill these out. Now, as I continue along, I'm going to notice that there are 4 boxes in total that I fill out. Again, I look left, I look up, so this one's going to be lowercase, lowercase. I look left, I look up, lowercase, lowercase. Now, this does not mean that these 2 parents are going to have 4 kids every time. Just for this one single trait, there are 4 possible outcomes. Now, what I want to do is I want to take that information and I want to do something with it. So over here, I'm going to write the possible genotypes that show up. Now, you could put the first genotype of capital F, capital F. This is a possible genotype for this trait, but there are none of them. None of these children here are capital F, capital F. So from a percentage point of view, that's at 0%, 0 of 4, or 0%. The next would be capital F, lowercase f, and I see that there is a capital F, lowercase f here, and there's a capital F, lowercase f here. So that's 2 of 4, or 50%. So I write in my 50% right here. My last possible genotype then would be the lowercase f, and again, I see that there are 2 of those. So lowercase f, 2 of 4, or 50%. The final thing is determining my phenotypes. The phenotypes are how they physically appear. So my 2 options for phenotypes are 5 toes and 6 toes. So 5 toes was my dominant trait, 6 toes is my recessive trait, and I want to know how many of these individuals in the children are 5-toed versus 6. So any individual that has a capital F will have 5 toes, and the only ones that have 6 toes would be those with lower cases. So these 2 right here both have a capital F, so they have one form of the dominant trait, so they will have 5 toes. So 2 of 4, or 50%. These 2 right here have the lowercase, so they would be the 6-toed, or 50%. All right, let's take a look at how to do a dihybrid cross. Now, in a dihybrid cross, we're looking at 2 traits at the same time. So in these crosses here, we're looking at traits found in guinea pigs, and the traits that we're going to look at are hair color and then the length of their ears. And so in my first parent, I've been told that it is heterozygous dominant for its brown hair. So heterozygous is 2 separate letters. So a capital and a lower, and then it's also heterozygous for its long ears, so 1 capital L, 1 lowercase l. So that's the genotype for parent 1. Parent 2 happens to be exactly the same, heterozygous for brown hair and heterozygous for long ears. Now from here I need to determine what the possible sex cells are going to look like. So it's important to remember that each parent will donate 1 copy of each trait to their children. So each parent's going to donate 1 B and 1 L. I just need to figure out which method or which sex cells I possibly could do. My preferred method is the foiling method, where I go with my first B and my first L, giving me capital B, capital L, which I'm writing over here on the side. Then outside, for the O and foil, the first capital B with the lowercase l. My inside would be my lowercase b with my capital L, and then the last would be my lowercase b with my lowercase l. If that method doesn't make sense to you, what you can remember is that there's 2 traits here, and one is the fur color and the second is the ear length. For this parent, we're looking at parent 2 now, so their traits are here. They have 2 options for the fur color, a capital B and a lowercase b, and then for the ear length they have a capital L and a lowercase l. So you can figure out all their possible combinations doing it this way too, saying that if parent 1 donates this first capital B, it could pair up with that capital L, giving you capital B, capital L. Or if parent 1 or parent 2 gives this capital B, it could pair up with that lowercase l, giving you capital B, lowercase l. The lowercase b could be donated as well, and that could pair up with that capital L, or the lowercase b could pair up with that lowercase l, and there's all 4 of your possible combinations. And you'll notice that I got the exact same results as I did by doing the foiling method. I just think the foiling method's a little bit faster. Now to fill out the Punnett square, everything inside of this 4x4 box here, these are the possible children that could result from these 2 parents. So what I do is I look left, so I look over to the left-hand side and I look up to fill this in. When I fill in the Punnett square, I always keep the letters together for the traits, then I go capital. So keep the B's together, keep the L's together, then go to the preference of capital. So I look left, I look up, so I have 2 capitals for B's and 1 capital B, 1 lowercase l, This would be capital B, lowercase b, capital L, capital L. For purposes of time, I've just jumped ahead and completed the whole rest of the Punnett square for us. So now I need to take the information from the Punnett square and get it down to the genotype ratios. The fastest way to do this is to just go one by one. So if I take my one that's in the uppermost, I give it a little check that I've counted it, I find it down here, and I give it a tally mark for 1. I find my next right here, I put a little tally mark. I come down here, I find it, I do a little mark here. I go through and I do the whole Punnett square by doing this counting method, just like this, so I get all of my completed genotype ratios. I've jumped ahead again now to show you the now completed genotype ratios after I've done all of my counting. To now get the phenotype ratios, the fastest way to do this is to take a look over here at the genotype ratios and find first which ones give me brown and long. And these 4 over here in this corner right here, all 4 of these give me brown fur with long ears. So I just take those numbers and I add them up. 1 plus 2 plus 2 plus 4. That number goes right here, which is 9. For figure out the brown and short, that's these ones right here. I add those two together, 2 plus 1, and I get 3 for brown and short. For then blonde and long, it's these 2 numbers right here. I add those 2 together. 2 plus 1 is 3. And then finally my last one is blonde and short. Add those together to get 1. All right, let's look at how to do an incomplete dominance Punnett square. What's important to remember with incomplete dominance is that neither trait here is dominant. And so when they're both together, when you have both alleles together, they mix to form an intermediate. So for example, if we're looking at tail length, the first genotype that's possible is LL, and that would give a long tail. Another genotype that's possible would be SS, and that would give a short tail. And then the final genotype that's possible is LS, and then we combine those together to form an intermediate, which would then be a medium length tail. And the same would be true for our other eye color, blue versus yellow and then green. So in our first Punnett square example here, we have a long-tailed female, which would be LL, mixed with a medium-tailed male, which would be LS. So then we have our same traditional setup for our Punnett square. Take my first parent, split their genes up along the left. Second parent, split their genes up along the top. Then I look left. I look up to fill in my Punnett squares just like this. So again, this information then gets transferred down to the genotype and phenotype ratio boxes. The only genotypes that show up for this cross are LL and LS. There are 2 that are LL, so 2 of 4 is 50%. And there are 2 that are LS, 2 of 4, 50%. So that means that my phenotypes then, LL gives me a long phenotype, and there's 2 of 4 of those at 50%. And there are 2 that are LS, which gives me a medium phenotype, which would be 2 of 4 or 50%. I don't need to include the SS at 0 and the short at 0 because they don't show up. You're welcome to include them if that makes it a little easier for you to remember all of your possible options. Here's a look at a codominance trait. Now in codominance, both traits are dominant. So in this case, if I have a capital R, that would give you, say, red flowers. Capital W would give you white flowers. And if you had one capital R and one capital W, this would give you like striped or spotted flowers where both colors show through. Neither trait is recessive to the other. They're both dominant, so in the phenotype, they both show through. In this cross, we have a red, white, striped female, so that'd be capital R, capital W. Crossed with a white male, that's capital W, capital W. Again, for the Punnett square, split up the genes of my parents along the side and along the top like I've done right there. Then I look left, I look up to fill in my Punnett square. Then I take this information, which I just got from my Punnett square, and I go to my genotype ratios. I see RW and 2 of those at 50%, and WW, 2 of those at 50%. That then means that I would have a, for striped would be at 50%. It was 2 of 4, and then white would be at 50% as well. The key with the codominance is I'm using capital letters and 2 separate letters to indicate that there's 2 separate alleles that are both dominant. Blood type Punnett squares are no different than any other type of the Punnett squares that we've used. It's just that we need to look at the possible genotypes for each individual for their types. So type A can be either AA or AO because A is dominant over O. B can be BB or BO because, again, B is dominant over O. O can only be OO. That's the only option. and AB can only be AB, remembering that A and B are codominant. So this is an example of multiple alleles where we have more than two. We have A, B, and O, where A and B are both dominant over O, and A and B are codominant. So in this problem here, this first one, I've been told that the mother is heterozygous for type A. Heterozygous means different. That means that the mom must be AO and not AA. If it was homozygous for A, then she'd be AA. The father is heterozygous for B, so same logic there, so dad has to be BO. I then take my parents' genotypes, I split them up on the side and along the top of my Punnett square, and I find out the possible genotypes of the children. This first child could be AB, we've got an AO as I look left here, a BO as I look bottom left, and then an OO for my bottom right. I then take that information and I put it into my genotype ratios box. AB are my possible, and AO, BO, and OO are what I see in the kids, and then how many of each. AB is at 1 of 4, so 25%. AO is 1 of 4, or 25%. BO is 1 of 4, or 25%. And then OO is 1 of 4, or 25%. Now my phenotype ratios are what they actually, the actual blood type that they have. So my first one would be type AB, and again, 1 of 4, or 25%. My second would be type A, and notice how I'm writing type A and I'm not writing type AO because type A is the actual blood type, 1 of 4 at 25%. My next would be type B, 1 of 4 at 25%, and then finally type O, 1 of 4 or 25%. For sex-linked traits, we're specifically looking at traits found on the X chromosome in these examples here. Now we're looking at fruit flies in this example, and fruit flies have either red eyes or white eyes, and red is dominant over white, and this is a trait found only on the X chromosome. So I've listed out for you here all of the possible genotypes for the females and also the possible genotypes for the males. Note that there's only two options for the males. Males are either dominant or recessive, and there's nothing on the Y because the Y chromosome does not carry the trait. I'm going to actually jump right to number 3, the Punnett square, to show you how to do one of these crosses. So in number 3, it says show the cross of a white-eyed female. Only way to have that would be X lowercase, because the white is the recessive trait. With a red-eyed male, that would be X capital R, Y. Again, noting that there's no letter attached to the Y because the Y chromosome does not carry the trait for eye color. I then split up the genes of each parent. I'm going to put the mother's genes over here on the left side, the father's genes along the top, splitting up the X and the Y. The children then, the boxes in purple, I look up, I look left to fill them in. What you'll notice right away as you fill out these Punnett squares is that half of the children should be females and half of the children should be males, and that should make sense because the male is either going to donate an X or a Y, which will then determine if the baby is a male or a female, because the females only ever can donate an X. They can't donate any other chromosome. So then my genotype ratios, again, I just look into my box here and I see that there is an X capital R, X lowercase r, and there's 2 of those, 1, 2 of those, so 50%, and then an X lowercase r, Y, and there's 2 of those at 50%. So the phenotypes then, I need to write out if they're male or female. So the first phenotype is a female with then red eyes, and the second phenotype that shows up here would be a male with white eyes. And again, my numbers should work out so that there's 50% male and 50% female. So in this case, there's only just females with red eyes at 50% because there's 2 of 4, and males with white eyes at 50%, 2 of 4. Let's look at how to solve a pedigree. And essentially a pedigree is just a family tree. So in these family trees, any square you see is a male, any circle you see is a female. Any individual that is shaded has whatever the trait is that we're tracing. So in this case, in this instance, the trait that we're tracing is a recessive trait. And so any individual that is shaded will have 2 lowercase alleles for that trait. So we can choose really any letter we want for this. Let's just go with the letter F. So this individual right here down the bottom has to have 2 lowercases because they are shaded in. So again, now I could find any individual that is shaded in, and I can go ahead and fill in the 2 lowercases for them. That then tells me that every other individual has to have at least 1 capital F because they are not shaded in and therefore mean that they are dominant for the trait, whatever this trait is. To solve the rest, though, what I like to do is I like to find an individual that's recessive, like this son right here, and then I like to work back to its parents. What I remember is that that child got one of their genes from each parent. So one of these lowercase f's came from dad, then the other lowercase f had to come from mom, which then tells me that both parents are capital lower. I know that they have a capital because they're not shaded, and I know that their second letter has to be a lower because they had a child, this son, that was lower. I'm going to use the same logic over here as I saw from this daughter right here. I know that this daughter got one of her lowercases from mom. The other lowercase had to then come from dad, meaning that dad must be capital lower. Sometimes people ask, well, how do I know that this daughter right here, how do I know that she didn't get both of her lowercases from mom? Well, because that's impossible. It's impossible for one parent to donate two traits to their child unless there's a genetic mistake there, which is incredibly, incredibly rare. So what happens here is this daughter got one of her letters from each parent. Now that I have the genes of both parents, let me get rid of these arrows so they're not confusing, I can then figure out the genes of this daughter right here. I know that this daughter has a capital because she's not shaded. And then if I look over here at mom over here only has lower cases, so every single time she has a kid, she's going to donate a lowercase. So that child there has to be capital lower. I'm going to use the same logic when I go from this right here, this child, to its parents. I know that its parents must both have a capital and a lower for the same logic that I used before. The lower must, from the two lowers from this daughter, must have one from each parent, just like that. Now here's a little bit of a trick. When I look at this son right here, I know this son's first letter is a capital because he's not shaded, but the second letter I can't prove with 100% certainty, so I put a dash in here. Let me show you why. Let's say that the dad donated the capital, right? Now that dad donated the capital, what could mom donate? Well, mom has one capital and one lower. We knew that from the last one that we figured out. So could mom donate the capital? Sure. Could she also have donated the lower and this problem would have worked out? Sure. So because this son does not have any kids, I don't have any further information. So both capital and capital lowercase are probable answers. So because I can't tell, I just put the dash. I'm going to use similar logic as I work my way over here to this son right here. I know that to go work back to his parents, that both parents have a capital because they're not shaded. And I know that one of them has a lowercase because this child has a lowercase. But where did it come from? Did it come from mom or did it come from dad? And because I don't have any information going up this way for the parents for these two individuals, I really can't prove it. I can't prove if it's dad that has the lowercase or if it's mom. So then both of them get dashes. To finish this problem off, over here, this son and this daughter also both have capitals because they're not shaded. The final part, again, is because I don't have the full complete information from their parents, I don't know what it is that this parent's donating here and what this parent's donating here. So both of these then also get dashes because I cannot prove it with 100% certainty. In this pedigree here, we're looking at sex-linked traits, so traits that are found on the X chromosome. I've given you in the bottom right-hand corner of this screen, we have all of the possible genotypes that could be shown for all of our individuals, male and female. I guess I would add an additional one here too for the females. A female could be X capital, X dash, and we'll get to an example of that in a minute. So when I solve these problems, what I'm going to do first is I'm going to find all of my individuals that are shaded, and I'm going to immediately fill those in because there's really not a whole lot of thought that's involved behind those. So like for example, this male down here has to be X lowercase and Y. It is an X and Y because it's a male, and the X chromosome is the only one that contains it. Because it's shaded, I know this individual is recessive. Same logic here for these two twin boys. They're X lowercase and Y. Over here I have another male, X lowercase and Y, and then finally, I have this female here who's X lowercase, and then finally my X lowercase and Y for this male at the top. Again, all I've done is I filled in all of my recessive individuals right away. They can be nothing else. The next thing that I can fill in right away as well is any male that's not shaded, because males only have two options, as you can see right down here in the box. They're either X capital NY or X lowercase ny. So any male that's not shaded has to be X capital Y. And so I can go through now and fill out every single one of my males that is not shaded. Now that I have those filled in, the only thing that's left are females that are not shaded. And so now for all of those females that are not shaded, I know that every single one of them has at least one capital letter in it. So all of the ones, if I go through, I could go ahead and write that in now. But again, what I like to do is I like to work my way backwards from an individual that I've already started with. So if I look at this male that's down here, which is X lowercase and Y, I can work back to my parents. What I remember is that the males get their Y chromosome from their dad, and their X chromosome must then come from their mom, which then tells me that this mom right here has to have one capital and one lowercase because she was not shaded to begin with. Now you'll notice that I already pre-shaded this one gray to show that she is a carrier. Oftentimes you'll see carriers half shaded like this. And now I just did it on gray because on Google Slides, when you do your assignments on these, you're going to change the fill color to gray. You can't do a half shade. So a carrier is, in this case, a woman that does not have whatever the trait it is that we're tracing. Let's say it's hemophilia, but she can pass it on to her kids. But the trick for solving these sex-linked ones is remembering who gets which chromosomes and from where. Males will get their Y chromosome from their dad always, and their X chromosome comes from their mom. A female will receive one X from her mom and one X from her dad. So if I look at these other daughters then that are down here, let me get rid of my arrow so I'm not confusing. If I look at, say, this daughter right here, I know for sure she's got a capital because she's not shaded. Then I need to figure out, well, what's her other one? Well, if I look at her dad only has a lowercase x. So that means that he will donate a lowercase to all of his daughters. Because if dad was donating a Y, then this would no longer be a girl, this would have been a boy. And so the dad only has one X chromosome. He will always donate his X to his daughters. So that gives me the same logic for this daughter here and this daughter over here. All of them have to be carriers. And again, to show those, I half shade them. So I'm half shading these carriers now. Let's take a look over here at the upper right-hand side of my screen. The only one I have left here to figure out is this parent right here. And there's several pieces of information that will then give me what this is. Let's take a look at this daughter. That will be the piece of information I'll use to solve this. I know that this daughter got one of her X chromosomes from her dad. That means her other X chromosome had to come from mom, which means that mom must be capital lower. And the reason I know she has a capital is because she was not shaded to begin with. That lowercase had to come from mom because look, they have a daughter that's two lowers right here. And so because she's two lowers, she had to receive one of her lowers from mom and another one of her lowers from dad. The final ones to fill out are over here on the left-hand side. I'm gonna use the same logic I used before in knowing that this son would have received a lowercase y from its dad, or just the y in general from its dad, which means the lowercase x had to come from mom. The other one from mom has to be a capital because she was not shaded to begin with. That tells me that mom must be a carrier. Last one to figure out is over here. This daughter starts, she for sure has an X capital because she's not shaded. Let's say that X capital came from dad. Well, mom has two options over here. Mom could have donated either one of her traits. She could have donated the capital, or she could have donated the lower, and this would have appeared as not shaded. So because I don't know for sure what it is, I'm gonna put in my dash here because I don't have any information about her kids, so I'm just be guessing. So X capital and X capital on both work, and X capital and X lowercase. So because I can't prove it, I put in a dash.